Okay, well, I think we're going to go ahead and get started. So I want to thank everybody coming to the Science in St. Louis seminar series. Today, we're going to hear from Melih Sener. His background is actually in theoretical physics and math. Yeah, about right. And he's really focusing now on using the very intellectual background, I would say, to better understand biological systems. So some of his research includes energy conversion processes in photosynthetic systems. At the protein, organelle, and cell level, assembly of supramolecular organization of multi-protein complexes and organelles, and lots of other good stuff. Some of my favorite work personally are his videos and models of photosynthetic organelles. And so there's a link on our website to his website, so where you can look at some of those videos. And so today we're going to hear more about this and the interplay of light and life envisioned by science, literature, and the arts. So thank you so much for coming.
Dan, if you could help me, maybe we should dim the lights just a bit. As my first act, I'm going to disappear, apparently. Oh, great, thanks. Okay, well, thanks for coming out here on this Saturday morning. I'll tell you a little bit about what I do, but I'll also tell you about what other people do related to what I'm very excited about, which is the way nature harvests sunlight. This is not only of academic interest, it is of perhaps of greater interest because energy, the economics of energy, is a very current topic, and maybe we can learn some lessons from nature. So that will be one of the themes that will be at the back of my head. Now, the title is a little bit of an overstatement. I should just come clean because I do science, but I don't know much about literature and even little about arts. But I am fascinated nonetheless that change and growth, as it is governed by sunlight, have fascinated humankind since pretty much we have recorded our history. And it's a theme that appears in mythology, in literature, in visual arts. And right now we can begin telling the stories of change and growth to such level of scrutiny, to such level of detail. We can basically take into account how individual atoms come together and exchange energy between them, and how light is harvested, one electron, one atom at a time. But it's still perhaps the continuation of the same basic curiosity. So with that in mind, my plan is I'm going to beg forgiveness from all the experts in the audience, and I'll show you what life looks like when you look at it at the level of individual atoms. So be patient with me. There are colleagues around you who are experts in these, and we are going to ask them to be patient with us while we look at how life, how proteins, how individual building blocks of all living matter is organized. And then we are going to use this as a background to look very closely at a machine that does photosynthesis. And lastly, pardon me for this, I'm going to have a look at how does it all work. And hopefully I'll convince you that out of this, we learn something useful at the end of the day. Okay, so as a prelude, so this is an appetizer of what I'm going to show you later. This is a molecular machine. What you see is just a colorful candy, perhaps at the moment. But a few minutes later, I'll tell you how to look at this with maybe a little bit more of a scrutinizing eye. So I said harvesting sunlight. The word that you have heard everywhere is photosynthesis. Photosynthesis is the harvesting of sunlight in nature. And usually what comes first to your mind is plants. This is not a part of a plant. It's part of a bacterium. So the kind of photosynthesis I'm going to talk about today is much more primitive than what you find in plants. And so on the right, you see sort of a zoom within a zoom view. At the bottom, there's a protein. That's where we are going to start from. At the middle is what you would call an organelle, which is to say a couple of dozen, a couple of hundred proteins come together and make a machine. At the top is a cell, which has many, many organelles. And we, as very complicated machines, have many, many, many cells. But my level of attention will never climb beyond that of a cell. And in fact, most of the time, I'll be talking about the level of details at the scale of a protein only. Wait, what is this doing here? I said that we are going to look at proteins, right? There are many ways to look at proteins. There are many ways you can scrutinize information that you obtain about nature. And whenever I think about presenting a new result, one of the first things that comes to my mind is, how do I parse this? How do I present it? There are many different ways you can look at information. And so Wallace Stevens apparently was pondering the same thing about, well, something he was obsessed about, blackbirds. I'm not going to read you much poetry, hopefully. I will resist the temptation. But I'm going to take his idea and throw it at proteins. So first of all, proteins are made of atoms. Well, everything is made of atoms, but specifically now you know just how many atoms and where are those atoms. What is their spatial relationship with respect to each other? Here is an atom. This is an atom. This is how big an atom actually is. So you see now how big a protein is when it is made of atoms. I'll tell you what this protein does in a moment, but let's go ahead and ask ourselves, oh, hold on a moment. Are you telling me that you can see individual atoms making up a protein? How do you know what this thing actually looks like? For most of what I'm going to show you, underneath there will be someone who has done a lot of very hard work to visualize, to capture data about what is in front of you. I'm a theoretician. I don't know how to grow bacteria. I don't know how to take their pictures. I don't know how to shine laser on them. What I can do is write equations on a piece of paper or compute their results with a computer. That's what I do. But before I can do any of this, somebody has to provide me, through their own hard work, the information about the whereabouts and the how-abouts of these basic building blocks of light. So in this case, this is a protein. It's going to be a light harvesting protein, as you will see in a moment. The structure of which is determined by Richard Cogdell and his colleagues through something called X-ray crystallography. It's a beautiful topic to read on if you would like to go ahead and have a look at it later on. So basically, you have amazing powers of observation available to you through which you are given information about these basic building blocks of life. And at every level of detail, a new method, a new experimental form of scrutiny makes more information available to you. And this is our lifeblood. We theoreticians are useless without experimental data. So even if I might not show you a specific paper and a specific name, every time you see a picture, think about it. Somewhere underneath that, there is information that came from somebody's experimental work. So atoms, it's not very useful to just look at something as a collection of atoms. Even if I make the distinction of saying, all right, in red you see the oxygens, and in blue you see the carbon atoms, and in white you have hydrogen atoms. This is just a big mess. It doesn't tell me anything useful. I should parse this information. I should try to aim for an economy of thought. Because we humans have evolved to deal with information through abstraction. We have evolved to deal with information through simplification whenever possible. So we want to find patterns. And for patterns, maybe one thing I want to do is look at only one kind of atoms. Let's say I only look at carbons. Carbon, we know, is the basis of life. It's abundant. Maybe I'll begin to see something if I look at carbons, but no luck. A lot of carbons, I'm not seeing anything useful. Maybe I should be more selective. Now I'm looking at carbons of a particular kind. These form the so-called backbone of the protein. Now we're beginning to see something come out of this. Next up, you can see that they actually form a chain. If you follow them up, they form a pattern. In fact, they make helices together. This is one of the familiar patterns. Whenever you see a picture of a protein, One of the common themes you see is helices. They also make sheets and various other structures. This is the kind of protein that has a lot of helices in it. So whenever you see a picture with a helix, what you should be thinking about is that there is a lot of atoms that are arranged in a way that their backbone forms a helix. So this is like a structural element. It's like a pillar that holds together a building. Now, well, okay, now I have accounted for a lot of the atoms. They form a nice backbone through which I get a helix. But is that all of them? Let me now show those atoms that don't belong to these helical structures. By the way, such things which are not part of the main part of the protein, we call them cofactors. That basically just means everything else. So cofactors, that's still a big mess. Maybe I can simplify that too in the same way that I have done before. First of all, I noticed that there are two kinds. Let's just call them the green kind and the orange kind. I'll tell you what they are in a moment. And I can simplify them a little bit more if you spend a little bit of time looking at what they are structured like. You find out that the green kind actually has these square-like atomic arrangements in the middle of which sits a happy magnesium atom. The other green atoms are actually sort of carbon atoms that surround the magnesium in a nice little square-like arrangement. And I clipped off the tails to not bother you with. They also have these sticking tails, which are important. But sometimes what you want is the abstraction. What you want is a simplification. So I have two different kinds of cofactors, two different kinds of ingredients. And the green one is called chlorophyll. Yes, when you go outside and see anything green anywhere, you're looking at these green pigments. Pigments because they are particularly selective in the way they react with light. And the orange ones are called carotenoids. I purposefully misspelled it here. They actually give carrots their orange color. So the green and the orange quite literally are the most abundant green and orange pigments that you're familiar with. So let's put the proteins back together maybe in a moment. So there's this actually famous saying, behind every great chlorophyll stands a carotenoid. This is a very common theme. What you find in all kinds of light harvesting molecules, you have carotenoids standing watch near their chlorophyll partners. And there is this very interesting photoprotection mechanism, which basically is a fancy word that says, if I shine light on you and you're not ready to deal with it, I'll come and relieve you of the burden. And this is done at an electronic level with very clever management of energy flow. And without this, without carotenoids, the system will burn itself immediately. So you don't want just something that harvests energy efficiently and quickly. You want it to last. So carotenoids make sure that your system lasts for a while. And so I can now put back the protein as a scaffold. Now I'm looking at it as a surface, yet another way to look at all this information. And maybe let us remind ourselves that in here, there's actually a helical arrangement. It's getting complicated again, but now I know what everything is. I have a surface representing the whole thing. I have a helix, which is my structural building blocks, and I have the two different kinds of pigments. And if I want, I can have a top view. Cool, now I know what I'm looking at. It has a name. It's called light harvesting complex 2. Incidentally, there's also a light harvesting complex 1 somewhere. So LH2 for short, and we are going to keep seeing this in many of the slides. I figured if I didn't do this, if I didn't go through what each of these structures look like, it would be very hard to know that underneath a picture like this, you have actually atoms. And we can't look at all of them at the same time. It's very hard to understand without a context how atoms come together and make life. This is life made out of atoms, simplified for the human mind. Okay, so wait a second, you're not really making much sense. All you have done is show pictures. How does it really work? Because if all I do as a theoretician is to simply make prettier pictures of the data that I get from somewhere, I can't really justify the reason of being here. So I have to do something useful about understanding how this protein works. What does it do? What is its purpose? And more importantly, why do you care? um, this is, I think, part of the dialogue between the little prince and the fox. If you have a mystery, you should not disobey it. And the mystery here actually involves quantum mechanics. So I told you that there are pigments. I showed you the chlorophylls and the carotenoids. What they do, what a pigment does, it captures light. Light arrives in the form of an energy packet called a photon. It's a wave, it's a particle. It dances like a wave, it hits like a particle. And when it hits a chlorophyll or a carotenoid, it creates an excitation. It's a fancy word. It should remind you of excitement. An excitation is basically just an energy packet, which is like a hot potato that has to be tossed elsewhere very quickly. There is a reason why you and I can't do photosynthesis. Because when normally light hits living matter, or any other matter for that matter, it decays very quickly. The energy is wasted. You know, you get a little bit of suntan when you stay underneath bright light, but you can't say, I'm not going to eat today, I'll just photosynthesize. You don't have the pigments for it. Some of you tried, it seems. And the way with which the energy is captured, momentarily stored, and delivered to other pigments is described through quantum mechanics. Quantum mechanics is the language with which you look at this energy packet, and everything you have heard about the counterintuitive nature of quantum mechanics plays a role here, because your energy packet is not in one place. We call it delocalized, which means that it's not in one place, it's everywhere at once. But just because we haven't evolved to perceive things quantum mechanically doesn't mean that there's a mystery here. We can still compute how fast that energy, even though it might be delocalized all over the place, jumps from one protein like this to the next protein. So we can talk about, you already see one of the neighboring proteins. So I have a protein that looks like this ring, and I have another one that looks like two rings that are jammed together. So that incidentally is LH1, light harvesting complex 1. So the neighboring protein, as it were. So I have many of these. Now let's have a look at them. Remember the cylindrical protein that we looked at? That is the green guys that you see here, the most abundant protein. If you were to open them up, you will see the chlorophylls and the carotenoids. Remember the 13 ways of looking at a blackboard? Now I have zoomed out. I don't want to look at all the atoms. It's too much information. I don't even want to see the pigments. I have literally about 3,000 of them. So now I have basically an assembly, for all intents and purposes, a device that's made of all these proteins, and they talk to each other using quantum mechanics. And if I want to describe this, one thing I can do is I can distill from the structural information what kind of a network these proteins form. A network, you should keep in mind, we deal with networks all the time. Our phones form networks. We deal with the internet. Information flows, energy flows through networks. And it is the same way that in these systems, you have a network of energy transfer. So chlorophylls and carotenoids talk directly to the chlorophylls, and chlorophylls talk to one another. Chlorophylls form an energy transfer network. And the means with which they are connected is this quantum mechanical description of the flow of energy. I promise to not show you equations in this talk. If you want to hear about them, we can talk about them later. So I love this quotation from Richard Cogdell. The beauty of how the purple bacteria, these are purple bacterial systems, so this is the system that he's talking about. The beauty of how the purple bacteria have evolved a solution to the problem of efficiently harvesting light energy comes from how they have, by trial and error, been able to elegantly exploit quantum mechanics. So in other words, nature has evolved sophisticated devices that know how to use quantum mechanics. And you can see their organization a little bit here. So I have pigments, they come together and make a basic building block. The building blocks come together and assemble into a protein. Proteins assemble into a larger machine, and then I have hundreds of these machines in a cell powering up the cell. For this bacterium. And how do I know what all of this looks like? Essentially, as I said, lots and lots of experimental data. This is like this proverbial story of an elephant in a room with a lot of blind people around it. I mean, I'm also one of the blind people, but other blind people tell me the little part that they deal with. And so we try to describe for one another how the trunk looks like, how the, all the other parts of the assembly looks like, essentially. And this is not only determining the structure, but also asking questions about how it all gets together and how it all interacts, how it functions as a machine, basically. So, well, a bird's eye view for all this is that this is essentially a means of capturing light and storing energy from the packet of energy that you receive from the light. So I have a solar collector. I have a battery. I'm going to charge the battery using the energy that I capture for the solar collector. So I'm going to show you a movie about how this is going to how this is achieved. Bear with me. This is the most complicated slide I have. Don't worry about the details. What this shows you is a process diagram. If you have a V8 engine, actually I have no idea what a V8 engine looks like, but I imagine they are popular, and I imagine that they make cars move, right? I looked up diagrams. They are complicated. And if you have a cell phone, if you look up their circuit diagrams, they are complicated. It's no different. There's a device, and it has a process diagram. It has components that talk to one another in different means. And this is no different here. And the movie that I'm going to show you also follows this. So we are going to basically show the path with which a packet of energy that is received from the sun by one of the pigments flows down these components, eventually making a deposit to a chemical energy storage unit, a battery. That we call ATP. You might have heard the word ATP. It's the universal energy currency of all life. Okay, so let's have a look at this all in a movie. And before I do this, credit where it's due, this is something that had been studied by and envisioned by my mentor, Klaus Schulden, for the last 4 decades, and about a dozen of us worked on making this movie. So like most things in science, this is a team effort. Okay, so down in a pond there lived a bacterium, right? I can't actually show everything in atomic detail, so I had to ask someone's help in making a drawing of this. But when you zoom in to the level of the individual cells, what you can see, now you're seeing a slice of a cell, and in the slice of a cell, you can see these compartments. There are hundreds of them. Now we're going to zoom into one of those. So the cell is full of vesicles, and each vesicle is put together from proteins. So let's have a close look. And you will permit me just a little bit of a Star Wars movie. It's impossible to do this without thinking Death Star. And parents, you'll have to explain your kids that, yeah, it's, to my great dismay, I realize that Star Wars references are no longer valid in the universe. So anyhow, this is sort of the Death Star trench run of this vesicle, where you get to zoom in to the individual proteins, and you can see what happens to one of the proteins as it captures light. Now, everything I show you is coming from experimental data, as I said, but occasionally I have to cheat. For example, I'm going to show you what happens to captured light, but neither you nor I have evolved the capacity to think of excitation, of energy moving around. So this is artist's imagination, as it were, of energy jumping from one chlorophyll to the other. In reality, it doesn't jump from one to the next to the next. Instead, what happens is that it jumps all over the place simultaneously in the usual quantum mechanical counterintuitive way that you hear elsewhere. Still, there's nothing confusing about it. It's just that we haven't evolved to deal with it. You can still compute what it does. Now, what happens in one protein is repeated for many. So you can see 3 neighbors of this LH2 that we have seen in the beginning. And you see in red something very similar that's called LH1. And in blue at the center is the so-called reaction center. The The reaction center has a purpose. It receives this energy, so it's like a game of hot potato, where it is delivered last to this catcher in the middle. And what the reaction center does is it converts this energy to an electric current. Now, here's a fun thing. When you talk about an electric current in biology, it's not like a copper wire with lots of electrons flowing. It's more like a carrier molecule will receive one electron and then happily will detach itself, swim elsewhere, deliver the electron, swim back, and wait for the next one. When I first seen this, I was practically giddy about it because you're kidding me. That's how electric currents work in biology. And this is a theme that you're going to see time and time again. You will see in the reaction center, there's basically a wiring diagram. I'm not going to tell you about how any of the details work, where this electron is delivered to a carrier, and the carrier sort of diffuses away and finds its next target, delivers the electron. But what I have done is I captured energy, and I use the energy to drive an electric current. It's still legitimate electric current. It's just that it's done one electron at a time. Well, actually, in this case, it's done two electrons at a time because the molecule can actually carry two in one go. So if I flip around to the other side, now I can see the next component. There's a new protein. I haven't told you yet what it does. It's essentially a converter. It shuffles your electric currents going one way for protons going the other way. So electrons are negatively charged, protons are positively charged. Basically, you shuffle them. By the way, why do you do this? Electrons are very finicky. You can't keep them in a place for long. Protons, on the other hand, you can keep them across the membrane for much longer. So this permits you to store energy a little bit more stably than you would otherwise. So basically, I traded with all this shuffling around for a little bit of stability. Let's skip this, actually. So at the end of the day, I need to close the circuit. All the electrons that were delivered to me, I give them back. There's another postal carrier molecule that swims back with the electron and says, fine, we have done whatever we needed to do with it. Here it is, take it back. And then the circuit starts again. At the end of the day, what I have done is pumped protons across the membrane. So this sphere is a membrane, right? And I have pumped protons into it. And these protons now want to come out because I have created a gradient. There's more of them inside than there are outside. And as they try to come outside, the only passage point that they are permitted to do is this gatekeeper called ATP synthase. The name should be a giveaway. It synthesizes ATP. So think of like essentially a washing machine, and it has sort of a rotatory component. And as the protons try to pass through the only path that they can, they generate a rotating motion at the bottom, which causes essentially some sort of stork rotation at the top. And what happens is that you have some kind of mechanical shuffle motion happening upstairs. So when I say laundry machine, I more or less actually mean it. You don't do money laundering, but you do energy laundering. You store it for a more permanent purpose. And so at the end of the day, I capture light, I drive an electric current, I stabilize it, and then I make ATP. And then ATP is something that you can basically store indefinitely. You have now achieved stable energy storage. You have made a battery that you can come back and use later on. And so if I look at all these components, essentially like a bunch of machine parts that come together and work well. So I have cooperation between these light-absorbing guys, which are in green, and then in red, similarly light-absorbing components. In blue is the reaction center, which does which drives the electric current. In purple is my transformer, the converter, which drives protons. And ATP synthase, at the end, converts these extra protons into the making of ATP. Great. So we have a machine. What do you really learn from this? Why do you care? Henceforth, I'll turn a deaf ear to prattling foliage with its arboreal metaphysics, its chlorophilosophy. I have no idea what he's talking about, actually. But I love that foliage prattles on and is trying to tell you something. What is it trying to tell me? What can I learn from this? One of the things that you can learn is actually, let's call them design principles of how machines in nature are built. And when I say design principles, I mean the physics-based functional principles of how these machines have evolved. So no theology today. Well, I lie. I'll do a little bit of theology later. But so basically, what are the functional principles with which these machines operate? And now I can put on a cap of that of an engineer. And I can ask, if I were to make a machine, let's say I have an invention that I wanted to be successful out in the market, there are a number of criteria that I should expect from it. It should be optimized to do whatever it is. It should be robust. That means it doesn't break very quickly. It should be reliable, and if possible, usually human-made machines aren't like this, but in nature, that's a luxury that nature affords. It should ideally be adaptive, meaning that when you change the conditions, it doesn't fall apart. It can deal with the new strain that you can throw at it. Now, it's possible to quantify each of these points and find many examples in nature. I'm going to only show you a few of my favorite ones. One, nature has evolved over time towards greater complexity. Like, one of the measures that you can imagine is important is, what is the packing density of stuff? If you look at an old Ford T model versus a new one, maybe one using a V8 engine. I have no idea if there are Fords that use V8 engines. But I imagine there's actually a greater complexity and greater packing density of components. This is what you see in nature as well. Primitive bacteria, like the one that I have been showing you about, has about 3 times lower packing density of pigments per, let's call it biostuff, without getting into the details, than what you find in plants or in cyanobacteria, which are these more complicated photosynthesis making organisms. So there's a drive towards complexity and optimization in nature. And there are some structural themes, like, for example, I told you that there's a lot of electric current flowing on. There are potential leak pathways where you can lose the electron that you're trying to deliver. And that means you have a competition between trying to put your chlorophylls as close to the reaction center as possible so you can give energy, but you want them further away so that your electric currents don't leak out. So there is what they call a no-fly zone around reaction centers. And this is a theme that you see in all kinds of photosynthetic organisms, from primitive bacteria to plants. And so these are not magic. They follow from the physics of the process. Physics of the process says, if you want to deliver energy, you want to be close. If you want to not deliver electrons by mistake, you want to be far. But the two processes have different spatial dependence on distance. So there's an optimal, nice distance where you can arrange everything and things will work. So nature knows how to do physics. And in the same way, there is usually some tolerance to the loss of components, some degree of robustness in the system that will keep the system from basically totally losing it if you were to hit it with a hammer. Forgive me for one technical slide. Maybe you can. One of the things that you can ask is, how optimal is this damn thing really? You can ask this question at the level of individual proteins, but you can also ask it at the level of the entire machine, at the level of the entire organelle. And one of the things that you learn is, so in this fanciful diagram, the darker the arrangement, the more energy output you get. And in the circle, you see where the particular figure that I've shown you, where the actual observed arrangement is. So this is, on the two axes are basically the number of components. Let's say I'm an engineer, I'm trying to dial the component list, right? And if I'm running a factory, I can say, okay, I want 10 of these machines and 20 of those machines, and there's a way to make sure that I'm getting the best turnout that I can. If I have too many of one kind and too little of the other, I'm wasting machine time. Now, what you expect intuitively is that the circle should be sitting at the top of where the image is darkest, but it isn't. And so that suggests that, wait a second, maybe nature isn't really as optimal as I expect it to be. Then you learn something important from a lesson like this, because nature doesn't care as much about optimizing one little ditty thing that you think is very important. It tries to survive. It has to have redundancy. It tries to have stability against strains that your poor little experimental dish in your comfortable laboratory might not have to deal with. So what we think is optimal may not be optimal for nature. So what else do you learn? Well, I told you that it is a machine. It makes ATP. It makes batteries. It is made by spending ATP. I mean, it's like, so I have a factory that makes me money, and I had to put down an initial down payment to produce it. So I can ask, what is the return on investment of this thing? Seriously, how much do I get in return for my initial investment? Or I can ask it in terms of time. How long does it take for this machine to pay itself back in terms of the initial investment of the cell, in terms of its energy cost? Let's have a quiz. Government bonds, what is the return on investment time? How long does it take for, I'm sure that some of you have investments somewhere. How good an investment is government bonds? Not very good, right? I think the number clocks at about something like 20 years. When you try to buy solar panels, one of the advertisement statements that you occasionally read is that solar panels are great, they pay you better than government bonds. What they try to say is that if you have a solar panel, you know, one of these big solar devices, you maybe like pay $200 to put it on your roof. They pay themselves back depending on where you live and how much sun you're getting. It's something like 5 years. How about this guy? About 10 to 15 hours, depending on how much light it is getting. That's how little time it takes for it to make enough ATP to pay itself back. So comparisons of efficiency, comparisons of optimization are hard to do. When you hear people talk about solar panels and photovoltaic systems, one of the things that you hear is that, oh, they're really efficient, and they do actually much better than natural systems. This is true. I mean, you can have, one of the measures of efficiency is if I capture a photon, if I capture a packet of light, how much of this, how likely is it that that energy can be used to drive an electric current? And for photovoltaic systems, for man-made solar devices, that number can actually be very high. You can get something like 50%. That's very nice, desirable efficiency for that kind of conversion. But it also depends on how you ask what you mean by that conversion. If you think about storing the energy in a chemical form so that you can reclaim it later, because you don't necessarily say, okay, you know, afternoon I'm getting some energy, but I better don't need any when it is dark. So you need to store it. Then the storage efficiency and the reclaim efficiency falls to something much lower. Still, photovoltaic devices do very well. But if you ask it this way, then the difference is colossal. The return that you get on your investment in nature has to be really competitive, because you are a bacterium, you divide or you die. And you better be dividing faster than your competitors. So evolution is a very highly motivating force in optimizing these processes. And you better do this in 10 to 15 hours or somewhere less than a day, because if you're going to take 5 years to divide, there will be somebody else that divides faster than you that will instead be filling the niche that you're trying to survive in. Why do we care? We have been fascinated by this for a very long time. I mean, our species, as long as we have recorded history, there are references to change and growth. Please permit me to read this, I love it. The one life within us and abroad, which meets all motion and becomes its soul, and light in sound, a sound like power in light, rhythm in all thought, and joy everywhere. I think it should have been impossible not to love all things in a world so filled. I don't know, I think he's talking about dancing, but maybe he's also talking about photosynthesis, and maybe the two are not that different from one another in a way. And this interplay of light and life and motion and rhythm, which, if you look at biology in detail, jumps at you. It has been there long before we had good microscopy. And in fact, the interplay between growth and change it's been there as a part of a lot of major religions. I mean, the ancient Egyptians were fascinated about how Ra basically provided Osiris with its light-giving rays. Now, actually, have a look. There are hands at the end of the rays. There are hands, life-delivering hands, and I think there's an Ankh somewhere in there. So in Hinduism, again, forgive me, I'm not trained in theology, but you find this appearing everywhere. I see thine eyes as the sun and the moon. I see thy face as a sacred fire that gives light and life to the whole universe, which apparently is from the Bhagavad Gita. And you see actually reflections of this in Abrahamic religions, even though they have been sort of purposefully excised in many of these cases, because competitive religions usually try to outdo one another in articulating these things. But so even today, we have a lot of fixation about mythology, even with all the science. I mean, so one of the things that you constantly hear about is that, hey, these are wonderful machines, and so there must be an intelligent designer behind them. But it turns out it's actually a very old argument. It goes down to Cicero with this, if you see a sundial, there must have been a purpose to have somebody have designed it. This is essentially a 2 millennia-old argument. The truth of the matter is, this is a philosophical question. I shouldn't have bias in the poetry I like. And I can't say, I like Samuel Taylor Coleridge, but not Cicero's argument, or somebody that says something from the perspective of a school of intelligent design. What I can do, however, is to say, let me scrutinize what my observations tell me. Because it's one thing to say, I choose to be inspired or not inspired by literature or arts. It's another thing to say, I will determine what is and what is not based on what I observe. And in that regard, you can again ask, so I jump back to the slide then, what does my observation about nature tell me? I have structure, I have function that I observe through these different species. And some are more complex than others. And either we are dealing with a very lazy God or a prankster. And this is actually one of the earliest references that I've seen to evolution in life. Well, phrased slightly differently, perhaps. The subtle life, the countless forms of living things, the wondrous tones of man and beast are full of strange astonishment and boundless change. And this is something that keeps happening in literature. I worked very hard to find a good example from music. I couldn't. If there are musicians in the audience, maybe you can talk to me later. But I'm pretty sure there are some references to photosynthesis in music as well. But seriously, what do we care? I mean, imagination doesn't get you bread. Imagination doesn't cure the sick. Imagination doesn't help you with your real-world problems. Actually, it does, but let's pretend that it doesn't. So what use is this? And one reason why you should care about it is that we are facing an energy crisis. We are greedy as a species. We want comfort, and we want it now, and we need a lot of energy for it. And the human civilization currently is using the energy equivalent of every single human being on planet having 2 electric ovens on, running all the time. I mean, it's a big number. Whatever a terawatt means, you don't have to worry about it. What you need to worry about is that it's a big number, but it's a small number compared to how much energy the sun is providing to Earth. It's about 1 part in 10,000 of the energy that is incident by sunlight on Earth. So if we were to convert even a small fraction of it, let's say you cover Earth's surface only 1% of it, and you have only a 1% conversion efficiency to capture that energy and convert it to human use, you're still talking about covering more or less the entirety of the current energy need of humankind. So maybe learning how nature harvests sunlight tells us, maybe it can tell us something useful about how we can answer our own energy crisis. Nature solved its own energy crisis about 3 billion years ago. It's still recycling the same old solution it did. Maybe it's about time that we learn something from it. And this is no joke. Prosperity correlates to energy use very directly. In third world countries, energy use per capita is an order of magnitude or less than energy use in developed countries. So I think one reason why we want to care about all this is maybe we can make machines, or maybe we can evolve sort of vat-grown examples that elaborate on what is seen in biology, biohybrid, bio-inspired solutions, as they call them, that can give us sort of maybe temporary reprieves in this energy crisis that we are facing. And I mean, essentially, what I cannot create, I do not understand. It's one thing to say, okay, fine, I know now how these components get together, but we are not yet quite at a point where we can successfully manipulate them to become a solution. We are at the beginning of doing this, and we are still years away from achieving this successfully. But ultimately, I think my own personal answer, why do we care? Because it's a mystery, and we cannot disobey it, basically. So on that note, I'm going to thank everybody. There's a long list of people that have been part of providing information for this. I'm going to basically especially thank Klaus Schulten, my mentor of many years, Neil Hunter, my primary experimental collaborator, a great many other people, and also a number of personal friends whose perspective I really appreciated. So on that note, thank you for your time. Nice. Pose questions are possible. Oh, well, thank you. Yes. Ah. That's the kind of thing that's easier to do on a computer than on a lab. I should probably bounce to... Oh, I'm sorry, yes. The question is, how do you address the issue of graceful degradation in a laboratory? To clarify for the rest of the audience, graceful degradation means pick out your cell phone, remove a random circuit from it. What are the chances that it will still work? It probably won't. Whereas in nature, usually you can remove components, and the chances are things will not die immediately. So graceful degradation refers to the kind of stability that you have in nature where, well, things degrade gracefully. And the question, I believe, refers to what can you do in a lab to test issues of graceful degradation? In my limited experience, this is the kind of question that is probably easier to ask computationally in some cases, although there are mutation studies that you can do, I imagine, to probe what happens to individual component loss. I feel like almost bouncing the question in the general direction of Dewey, but it's... Well, okay, so here's the thing. If I'm making a computation involving, I have 100 chlorophylls that are playing hot potato throwing energy to each other. I can easily ask the question of, let me cut this one there. Whereas if this was a lab experiment, I have to worry about removing the binding component that would remove this chlorophyll, and then worry about, would this thing still assemble and work so that I am only looking at the loss of this one component and not everything else? That might be hard to do as an experiment. Whereas some of these things are perhaps easier to do computationally. This is a very hard question. There's no easy answer. I haven't given a good answer for myself. Neither have I seen a consistent answer given on this in the literature. I think this is one of the ongoing quests. Anyone else? Yeah.
I wanted to show that movie to someone. Where could I find it?
Oh, I think we are going to post it to YouTube somewhere. I mean, this lecture itself will be available on the internet, as I understand. But the movie itself, I think we are making available on YouTube somewhere. Let's talk about it afterward. If you give me your email, I'll try to look it up and send it to you. Thank you.
More questions? So, what made you pick this organelle to look at modeling the generation of ATP?
Because data was available. Currently, I'm very excited at doing the same thing for the plant counterpart because data is now available for that. But it's we theoreticians have to be opportunists in terms of what are good, outstanding problems for which new data has become available. So you have to address in a timely manner whatever new puzzles have come out. Yeah.
On the same sort of question, then you said you were wanting to look at the plant counterparts. That's photosystem 1 and 2. I'm assuming it's
At the protein level, photosystems 1 and 2 would
okay.
be the primary proteins in light harvesting plants. Yes.
So the whole time I'm trying to compare and contrast what I know about that. No, I know more about that than I do about bacterial photosystems. And one of the things that struck me is how physically close and connected the two photosystems here are, the light harvesting systems, and plants, they're more separated, and you've got like ubiquinone shuttling and stuff like that. So how, I mean, what kind of compares and contrasts have you seen so far in doing that work?
That again is one of these questions that can be elaborated upon for hours. Long story short, what you find in bacteria are sort of a looser, less focused arrangement with greater distances between components and a smaller packing density, and less opportunities for, let's just call it vertical development, which in some cases is almost quite literally the case. In plants, they huddle together closer, and it's sort of like comparing, I don't know, maybe a Champaign-Urbana to New York City. You have skyscrapers of light harvesting components stacked on top of each other, densely packed with a lot of traffic buzzing between. And not only that, but these skyscrapers get disassembled one morning and get reassembled on New Jersey the next day. I mean, that's roughly what it looks like in plants.
Like the phycobilisomes?
Other questions from the back, please. I was just pushing. Yes. Beautiful question. I
So just
love
for
it
the
when I
people
get asked
online,
this.
the question was, how long does the purple bacteria live?
Oh, yes, thank you. I should have bounced this out. Okay, so let me explain why this is a very good question. So the question that I got asked is, if it takes, let's say, somewhere between 8 to 15 hours to make enough energy to pay back the energy investment for making one of these machines, doesn't it supposed to tell me something about how long the bacteria lives? First of all, does the bacteria have even enough time to make one of these things? Maybe it divides so quickly that this is just not a feasible way. Turns out that one of the best ways to test this is you dim the lights and you make sure that there is no nutrients available to the bacteria. Basically, you starve them. You torture them to the point that their lives depend on growing as many of these things as possible. Quite literally, their lives depend on making this stuff. Under such conditions, they basically go haywire and fill the cell entirely with these vesicles. Then, if you think about it, the doubling time of the cell, which is to say to make enough copies of this as the ones that you have started, roughly, because the rest is just a bag to cover everything. The doubling time in such a case should correspond roughly to how much time it should take you to pay back the energy cost. And then the two numbers compare. Now, I have to admit that this computation that I told you is a crude one. And this is the kind of thing that will take years to get right with numerical accuracy more than, say, a factor 2. But Oh, these are hard questions. If I tell you that you can guess it within a factor 2, that's actually a good thing. That's the kind of thing that actually makes the theoretical computational scientists happy. That you are, without using free parameters, without using any gimmicks, you estimate something and it's in the right ballpark. But it will take us a very long time to actually account for all of the details for this, let's call it the energy economics of the cell. That's not something that we can do very quickly. But the two numbers compare. The doubling time under very dim light is comparable to the time for the return of investment in terms of energy of these vesicles for such dim light conditions. Did you have another question, or did I misunderstand that?
More questions? You? Yeah. No. Okay. Well, let's thank our speaker for a great talk.